Welcome to Trial Site News podcast series. Today we have the pleasure of introducing Dr. Bruce Patterson, a decorated scientist and well-known physician, as well as CEO and co-founder of InCelldx. Now, Dr. Patterson was trained in medicine in Northwestern University Medical School. He was actively involved in research during the early stages of the HIV/AIDS epidemic, which, back then, Dr. Patterson investigated cellular reservoirs of HIV-1 using molecular and in situ technology patented in his laboratory. During his residency, he determined that enough HIV virus was present in infected individuals to account for massive destruction of the immune system. This work was paradigm altering and led to a study established in Science in 1992. Now, Dr. Patterson went on to be named chief resident of pathology at Northwestern Memorial Hospital and has authored over 100 manuscripts and book chapters. He has also served as medical director of diagnostic virology for Stanford University hospitals and clinics. So with all that being said, thank you for joining us, Dr. Patterson, and welcome to the show.
My pleasure. Happy to be here.
The pleasure is ours. Now, I'm going to jump right into this today. We obviously face an unprecedented situation in the modern era. Trial Site News' focus is on research and promising investigational therapies that are still in the research pipeline. COVID-19 has been incredibly difficult as it's a brand new pathogen and we know so little about it. Would you agree with this assessment?
Absolutely. This is an emerging pathogen, and it caught us by surprise. But it's not unlike the early days of HIV where we didn't know what it was, and so it's not unusual from my perspective to see a lot of HIV researchers crossing over into COVID because clearly there is virologic aspects of COVID as well as immunologic aspects of COVID. So they're not that different. And for those of us who have been through other pandemics like HIV and H1N1, this isn't unusual. It's a lot of work, but we're facing some of the same challenges that we did in the early days of HIV. First with diagnostics. Diagnostics were a huge challenge in HIV, and then of course trying to figure out how the virus did its damage as we are with COVID right now was the next big challenge, and then ultimately how to either treat it or come up with vaccines or therapeutic neutralizing antibodies.
Yes, well, a lot of similarities between the two.
Mm-hmm.
Now, there has been several $1 billion invested thus far in vaccine development, with a number of candidates progressed to phase 3. Now, given that billions of dollars were invested in vaccine development for HIV/AIDS and none was ever found, what are your thoughts on the prospect for effective vaccines in the short and intermediate run with COVID-19?
Well, again, I think vaccinology has matured and, of course, evolved over the years, but we face similar challenges. We face a virus that has significant sequence variation, and we're going through the same types of paths to vaccine development that we did with HIV. First, is it safe? Are you going to get the virus, or are you going to get a significant reaction to the vaccine? And that's obviously phase 1. And just like in HIV, they would call out these victories, for HIV vaccine, exciting new results coming out of X, Y, and Z. And frankly, that was just phase 1, and we showed that it was safe, or you didn't get HIV from giving the vaccine. So hurdle number 1, and there's really 3 hurdles. There's safety. Does it elicit an immune response? And is that immune response protective? And it's those last 2 hurdles that are absolutely critical and still to date are relatively unknown. I mean, we know that some of the vaccine candidates elicit an immune response, but by the same token, we're hearing that antibody protection in patients who have been infected and have a natural immunity only lasts for 72 days. So there's a lot of contradictions that you hear through the media that I've been picking up because I've been through this before. But the layperson can't readily pick up some of the contradictions that are out there. I mean, to me, sequence variation, vaccine, neutralizing antibodies with sequence variation, HIV, look at hepatitis C. Hepatitis C, we've cured it with therapeutics, but there's never been a vaccine for hepatitis C, which also has a very high mutation rate.
So then let me pick your brain for a bit then. What's your general outlook on COVID-19 and why do you think there is such a variation in the virus?
Well, that's inherent in some viruses versus other viruses. And where there's been successful vaccines, it's been in viruses that have very little sequence variation, and you can build a vaccine that across the board is effective. I mean, measles is the best example of a vaccine that's close to 100% effective. And obviously, we've almost eradicated polio through vaccines. So, you know, there are great successes in vaccines, and I think when you see a less than optimal vaccine, it's because of tremendous sequence variation of the virus. You know, there's different reasons for that. HIV had basically had an enzyme called reverse transcriptase that had really poor fidelity. In other words, it was like a Xerox machine that was really bad, right? So if you imagine a Xerox machine that every couple of letters in a word, it put in something different. That's sort of the analogy in HIV vaccinology. So that's something that's we have to contend with, not for the same reasons in COVID, but nevertheless is a challenge for all the vaccinologists.
Right. So what about long haulers then? Is this a growing trend among COVID patients?
So long haulers is something that we've been tremendously focused on lately. And, I tend to divide COVID into 4 phases. I mean, there's an early phase that's virologic. You can detect the virus. There's no symptoms. As a pathologist and a virologist, when you tell me there's symptoms, that's an indication to me that we're starting to express these inflammatory proteins called cytokines. So for instance, interleukin-1 causes fevers. Interleukin-6 and interleukin-8 cause muscle aches, joint pain. So when you have, quote, symptoms like those, that's telling me that the immune system is kicking in. And we're in another phase, which is sort of the virologic phase with early immunologic phase. Then we get into a phase, if you get into the moderate, severe, even critical patients where, you're still in the virologic phase, there's still virus, it's still replicating, but you have a profound immune response. And then last is the long haulers where, months after infection, they've gotten over the hump. They're not in, harm's way, but they're suffering from persistent symptoms. It's taken its toll both physically and mentally in terms of well-being. And that's a different phase. The virus is gone, right? This isn't a virus that has a latent phase like an HIV or some of the herpes viruses that stay in your body for years and come out at certain times. The virus is gone, but we're left with residual inflammation, residual inflammatory changes that through our long hauler studies have been incredibly heterogeneous. So there again, the reliance and the importance of diagnostics, and that's what we're doing here at InCelldx, to look at these long haulers and apply personalized medicine, just like we do in cancer, to these long haulers, because every single one is different. When I look at patterns on immune profiling and patterns of residual cytokines that remain elevated relative to normal, you're seeing incredible heterogeneity that is going to require different therapies for different patients. And you know what? That's exactly what we're doing in cancer as we tailor therapies to the specific tumor. We're going to be tailoring long hauler therapies to the specific patient. And we're in the midst of forming a chronic COVID treatment center with some partnerships with telemedicine companies here in Silicon Valley that will allow us to bring together very sophisticated diagnostics, which we've launched through our partners at MD Biosciences, with physicians who are, including myself, that can interpret the results of those tests, come up with treatment regimens, but also pull in a group out here in California that can help with some of the mental health and the behavioral aspects of what these patients are going through. So we can have a true soup to nuts treatment of the individual. And by personalized medicine, by treatment, I don't mean from the neck down. I mean complete treatment, because these patients are really suffering. They've been suffering for a long time. They have some depression, which has been related to a cytokine called TNF alpha. But altogether, we want to treat the whole person and get them on the road to recovery ultimately after COVID and these lingering effects.
So what then would be your perspective on the challenges that we are facing with containing this pathogen? Does it make sense to keep much activity shut down, or would there be a more regional focused approaches where there's hotspots of shutdowns, et cetera? Now, there is concern that COVID-19, like HIV, is like HIV in that it mutates and many are asymptomatic. What are your thoughts on this?
Yeah, I mean, that was the big challenge in HIV is you didn't know you had it, and was a huge challenge in terms of transmission. And next thing you know, we had 40 million cases. So that is a problem. And and testing. I'm not going to say testing is a problem. I think people, if you're trained in laboratory medicine like I've been, you realize that tests are going to have false positives and false negatives. I mean, we hear it every day. If you listen to sports, Indianapolis Colts had a positive, they shut everything down. Then the next day they found out it's a false positive. Right. Or, a group of people have false negatives. I mean, that's just, that's part and parcel to every diagnostic test. That should be the lesson to, the world, is that every single test that is performed in medicine has a performance metric associated with it: sensitivity, specificity, false positives, false negatives. It's extremely rare, if it even happens, that there's a test that is 100% sensitive and 100% specific, meaning there's no false positives and there's no false negatives. It just doesn't happen. And so if it did, there wouldn't be pathologists. So there wouldn't even be a specialty for me to go into, 25, 30 years ago. So, but it's a good lesson. And the other lesson is, you'll hear companies talk about, well, 100% sensitive and 98% specific. Well, there's a big difference between analytical sensitivity and specificity and clinical sensitivity and specificity. One is you use this diagnostic test on a laboratory sample and it does exactly what it's supposed to do, and it's close to 100%. Problem is when you get into a patient, taking the sample and what happens to the sample has to be integrated into performance of the actual test. We always used to say you could be NASA on the back end and have the most unbelievable test known to man, but if that nose swab sits in a parking lot for 2.5 days, what are you going to get out of it, right? And we know that doesn't happen, but nevertheless, we still have taking of the sample, how much of the sample do you get? What's the viral load? There's a whole lot of variables before it even reaches one of these great sophisticated machines by these great diagnostic companies. So much goes on before it even gets there that introduces false negatives and false positives. And again, that's been an issue we've had to deal with in terms of saying, triaging patients. And when I was running virology at Stanford, we ran into the same issues. And people love the so-called rapid antigens, which more or less are like a pregnancy test, where you put something on a strip and in 5-10 minutes you have a result. Yeah, they're great. They're very fast. The problem is they're not very sensitive and they're not very specific. By not very, I mean, less than 90% sensitive, less than 90% specific if you looked at some of the rapid virology tests from the past. And that made it that made it really difficult in order to triage patients. Do you treat them? Do you send them home? Do you put them in a hospital room with other infected people when it could be a false positive? These were all considerations that are very difficult to manage. And that's what we're facing today with all the testing. Now, rapid testing is great because it's rapid and you could literally do it every single day, which I always bring up the example of the Pap smear. The Pap smear has saved millions and millions of women's lives since it was introduced in 1949, but it has a sensitivity of about 55 to 60% for pre-cervical cancer, which is terrible when you compare it to HPV testing and some other things. However, because you did it every year and the process was relatively slow, it became very effective. So there's strategies you could deploy even with rapid tests with less than perfect performance metrics to do the tests over and over and over again on the same person. Again, then it becomes an economic issue. The NFL can afford it. Schools that I consult with across the United States can't afford to test every single student every single day using a rapid test. So these are some of the real challenges we face in how do we manage infection, aside from saying, wear your mask, socially distance, and stay at home, which unfortunately is probably the best way to keep the transmission rates down.
Now, as far as the emerging treatment landscape, could you share your perspective on what are promising investigational therapies based on situation? So what could possibly work for early stage inhibition of the virus versus what might work better for more severe cases? For example, remdesivir is approved under EUA, but it can only be administered intravenously in the hospital.
Yeah, so there's a lot of different variables that go into the therapeutics and what's being tested right now. And you mentioned one of them, obviously, the delivery route. Is it a pill? Is it a sub-Q injection? Is it IV? That's going to have great impact on the different stage where you can deliver it. I mean, you can't have an outpatient with viral infection with no symptoms and give them the IV. I mean, that's just not going to happen. So there's that consideration. The other thing is you have to divide the therapeutics as I described in dividing the continuum of COVID disease. So the early stages, asymptomatic virologic. What's going to work there? Well, not the immunologic modulators like leronlimab, maraviroc, the Humanigen drug, and RLF 100. Things that affect the immune system come into play, as you get past that mild stage of COVID infection. So early on, ideally, it would be, an antiviral, and you're probably only going to give it if they start to have symptoms. But when they start to have symptoms, it's the early parts of the immunologic phase. So, people have talked about combinations or drugs that could accomplish both by reducing the amount of virus or the viral load, and also preventing production of cytokines by the various immune cells. In the middle of the disease is the most difficult, because that's when the patients are truly symptomatic. A lot of them get hospitalized at moderate infections. Most of them require supplemental oxygen. And that's where you have this battle between the virus and the immune system. So whatever you give, you have to make sure that it doesn't affect one or the other. So solely an antiviral, when you're having the beginnings of the cytokine storm, it's questionable whether or not that's going to be truly effective, because the immune system's kicking in with this chaos that I described, back in, March and early April. And then you have to start considering, a more immune-based approaches, like CCR5 inhibitors as a class. And we're now, working with 3 different ones, because we identified CCR5 as a target for COVID early on in the disease. And in fact, now that 4 companies are involved, we've pretty much justified that that's an important target in COVID. And then there's other ones, GM-CSF inhibitors, et cetera, that target the immune response. So number one, and again, I think it all depends on diagnostics. You have to determine where are you in the spectrum of COVID disease. And then I think therapies are going to depend on where you are in that spectrum. And that spectrum includes long haulers, where it's long haulers, no virus, residual immune and inflammation. And then there's the middle 2, which are difficult because you have viral replication and you have a chaotic immune response with so-called cytokine storm. And in the early phase, where you just have asymptomatic viral positive individuals. But therapy is going to have to be tailored to those different stages. And again, InCelldx is developing the tools to be able to determine where you are in that spectrum of disease, and then help being able to monitor, the disease, as they're treated.
So what about the work that you did on leronlimab? Does it hold promise?
Well, again, I think now we are talking about the whole class of CCR5 inhibitors, because leronlimab brought others to the table. Pfizer's Maraviroc, Orion Biotechnologies, OB002, both of which we're working with, and then Sencrevirac by Takeda, which is both CCR5 and CCR2. Is now getting some publications and some press, and apparently is being used in a trial by the NIH. So there's more players now, and again, I think it's just support on what we did early on by identifying the mechanism of action that would be effective for COVID, which is repolarize the macrophages, keep them from making IL-6. Mitigate the mass migration of immune cells to the lung that was resulting in severe depletion of or severe low levels of CD8 T cells, which are required for antiviral responses. And again, when we restored those with leronlimab, we were able to see plasma viral loads go down to undetectable by 14 days. So, again, there's always the option, and CCR5 may be the target, where you can modify and modulate the immune response, get an effective immune response, and also take care of the virology, instead of having always to turn towards, I need an antiviral and I need a immune modulator or even immune suppressant. People are doing lately with using steroids and antivirals. Now, that's all well and good. We do it for shingles, but we had a very strong antiviral acyclovir class of drugs for shingles, and then we'd use steroids. And again, whether or not we have an effective antiviral, it remains to be seen, to be used in conjunction with a steroid. I've always been nervous about using steroids while you still have viral replication, because it causes generalized immune suppression, which could make you more susceptible to other infections besides COVID as well. So, there's a lot that needs to go into treatment strategies. But I think our early work with leronlimab really identified a pathway that has proven successful for treatment of COVID, and we're awaiting more clinical trial data soon. But then again, like I said, the fact that 3 other companies are in the game with the same target is good justification that we were on the right path way back in February.
So let's shift gears a little bit. I want to talk about monoclonal antibodies that President Trump is touting, and then I want to talk about ivermectin. So let's start with the monoclonal antibodies. Do you think that these could become commonplace and economical treatments if they are ultimately approved?
Well, again, you have to clarify monoclonal antibodies, because I know when people talk about monoclonal antibodies, they're talking about neutralizing antibodies, antiviral monoclonal antibodies. But for instance, leronlimab is actually a monoclonal antibody, but it's an immune modulator. So, but I think every time someone says monoclonal antibody, they think, someone's mentioning leronlimab. But I think the key is to, number one, to think of monoclonal antibodies as just a type of drug class. What it actually does is what needs to be considered. So there's neutralizing monoclonal antibodies. It's an exciting opportunity. Like I said, if we could come up with something that worked with neutralizing antibodies, I still have my reservations because of sequence variation. Some of them were designed in the spring, and by now there's going to be sequence differences in the infections that are out in the community. The other thing is, how effective are they going to be over time? Again, with the natural history finding that antibodies against the virus in individual patients aren't even protective after 72 days. And so again, I think they've shown great promise, like I said, in a flask, and they're showing in some studies to be somewhat effective in patients. There's a couple of safety issues that have popped up as well. So, it still remains to be seen, how effective neutralizing antibodies are going to be as a class of antivirals. But that'll be a class of antivirals in addition to other antiviral drugs. So, it's part of our armamentarium, and I think they're still in play.
Right. Now, Trial Site News has chronicled the use of ivermectin in many countries, and there seem to be growing observational data that it may help inhibit COVID-19. Any thoughts on this approach?
If you follow the path of all these things that pop up, it's one and the same. I mean, they show that it works in a flask on cell lines, against cell lines infected with virus. They use the drug and it's effective in a flask. Then they go to patients, and then, again, they haven't done the double-blind placebo-controlled trials yet, but they have anecdotal data from various studies. And, at this point in a pandemic, you don't ever want to rule anything out. But, I think you have to be cautious about how you interpret that. I always tell a story, and friends and family around me hate it when I tell it, but back in 1991, I used colloidal gold in culture with HIV, and it stopped it cold. And I was really excited. I thought I had the answer to, treating HIV. And colloidal gold was a drug that was used for arthritis, so it was FDA approved. You could use it in patients. We put it into HIV patients, and it did nothing. So, that was a lesson learned for me 30 years ago about how to, that big stretch between something working in a flask and something working in a patient. especially in very heterogeneous diseases like COVID, I always have to take, with a grain of salt.
Now let's shift over to InCelldx. It's a very fascinating company. Now, it was founded in 2009, and we know that it is a single-cell diagnostic company committed to advancing precision medicine by offering transformative diagnostic and prognostic clinical patient information based on an innovative tech platform. Can you share with the audience how you got this company started and what idea got you going?
Well, again, I think it was, it started at way back in the work I was doing in HIV, and obviously developed a lot of technology there where we actually were able to find where HIV was hiding, which was the big question back in the day, by being able to do molecular biology in cells and in tissues without destroying the cells and tissues. Over the years, things have evolved. There's lots about, next gen sequencing and single cell analysis where, they put, a tumor in a blender and then look for 600 different markers, which is fine to answer the question, yes or no, is a mutation that's going to correspond to a drug, is it present? The problem is you've lost the context of the cells and tissues. That causes an issue when you think about tumors and talk, again, another conundrum. In one breath, you're talking about tumor heterogeneity, meaning within the tumor, there are different tumor cell clones, different tumor cells that genetically are different from other tumor cells. So they're different, but you're putting everything in a blender and you're getting a composite of what's in that tumor. InCelldx has been committed to keeping the cell intact so that you can look at the full spectrum of mutations and markers that correspond to therapies on a cell-by-cell basis. And when you're talking about heterogeneous disease, that's where we shine because we've developed the technology. There is not a molecular biology technique we cannot do in the cell. We've done in situ hybridization, in situ PCR, real-time PCR, in situ, and we just announced a deal with Factorial DX, where we're actually doing in situ library prep for sequencing. So we can unequivocally identify a cell with multiple antibodies, do the library prep within the cell so that we know what cell it is coming from, and then eventually lyse the cell and do the sequencing component. That's what we do. And again, in the last 5 years, we've gotten involved in immuno-oncology with PD-L1 and checkpoint inhibitors. I categorize CCR5 in that bucket now. In 2016, we patented targeting CCR5 on T regulatory cells, which inhibits the immune response against tumors. So that's a very elegant approach to enhance the effects of PD-L1 inhibitors or PD-1 inhibitors in cancer. And then, of course, we were just granted a patent for PD-L1 as a diagnostic by flow cytometry and in circulating tumor cells, which is very readily used to determine if you're a candidate for these very sophisticated and successful immunotherapies. So, I think we've evolved. We began in virology, we morphed into cancer, now we're into immuno-oncology, and then we've taken what we've learned in cancer and immuno-oncology, and now are back attacking the virus, which uses the immune system response to viruses is very similar to the immune response to cancers. And so we're rolling that into our diagnostics and how we view disease and how we view how disease can be treated. And along those lines, when we've had the opportunity, when we've identified new targets, either in cancer or in virology, InCelldx as patented targets for various pharmaceuticals. So that's been our strategy. It's evolved over the years, and it's a really exciting time at InCelldx right now.
Now, can you expand on what InCelldx is doing to contribute to fighting this pandemic?
Yeah, so again, we signed a deal with MD Biosciences to launch very sophisticated immunology diagnostics around the globe through their network of labs. And we're also putting together with a variety of partnerships a long hauler diagnosis, treatment, and monitoring initiative or company, as you will, that will be made up of all the components from diagnosis, treatment, monitoring, counseling, much like genetic, a lot of the genetic market where you have companies doing, various genetic tests, and then they go see their genetic counselor to discuss how to manage, the results of the genetic testing. We're putting together a basically a initiative with a number of different companies to provide, basically a long hauler institute and treatment center where they can go and get them on the road to recovery. Because, like a lot of chronic diseases, they'll go to, various physicians and they'll say, oh, you'll eventually get better, or just wait it out, or gut it out, or, I don't know what we can do. We want to be better than nothing. So that's what we're doing, and that's what we're basically at the beginning of putting together right now.
So, long-term vision for InCelldx. How do you see the company's business prospects? Does the company seek more financing?
Well, the good news about InCelldx is that we've run lean. We have, over 90% margins on all of our assays, which allows us to realize, cash flow positive and profitability in years. And, and I think we want to grow organically. We have 14 diagnostic tests and 14, different assays for different cancers, viruses, it's immuno-oncology, et cetera. We're going to take advantage of our extensive patent portfolio of over 87 patents. And, we're going to build it organically. So, we haven't done, enormous raises to get to where we want to go. And so what we've done is we've done strategic deals and lived off that for 10 years. And now we're at an inflection point where we have, some opportunities to really get to that next level in terms of revenue and growth.
So, finally today, last question for you. The Trial Site Network includes potential partners as well as investors. What message would you like to leave them with in reference to InCelldx?
Well, I think follow us, see what we're doing, call us. The great thing about InCelldx, and the thing that's kept me just captivated for 10 years, is that every week is different. Like I said, with a portfolio of 87 patents, obviously we're very active. We publish 2 to 4 papers per year. And I think now, we've nicely built out all of our products, and we're really in a mode where we're selling, and going with what we have. I think, we'll continue to innovate, but I think the exciting thing about InCelldx is it's always different. So it's something you have to keep an eye on, because next week we could be doing something with our extensive portfolio that's novel and necessary, and much like we did with COVID. I mean, we were able to respond to COVID. After I went to China in mid-January, talked to our 3 partner companies in China, our customers, essentially, we were able to respond very quickly to what we were hearing. That was an IL-6 disease, and started developing cytokine assays, and found some other things in COVID, like the dependence on RANTES and CCR5. So it's been a wild ride, but I think, like I said, the fun thing about InCelldx is it's always got something going on. It's always different every week.
Well, thank you so much for taking the time for joining us today, Dr. Patterson. We appreciate you speaking with us.
It's my pleasure. Thank you so much.